organs, tissues, cells, and cell products that differentiate self from the non-self neutralize potential pathogen organisms. Immune system consists of innate and the acquired immunity. Mainly immunoglobulins are formed by B lymphocytes to plasma cells. Immunoglobulins are mainly composed of light and heavy polypeptide chain. Most are combination of two light chains and the two heavy chains. What is the difference between the antigen and antibody? Antibody is also called as immunoglobulin. Antibodies are Y-shaped molecule. Y-shaped molecules are protein manufactured by the body that help the fight against the foreign substance, and that is called the antigens. Antigens, any substance that stimulate the immune system, and they are produce the antibodies. Antigen can be bacteria, virus, fungi that can cause infection or some diseases. What is mean by antibody and what is immunoglobulins? Actually, antibodies are a specific glycoproteins configuration, which is mainly produced by the B lymphocytes and plasma cells in response to specific antigen and capable of reacting with that antigen. Whereas all antibodies are immunoglobulins. Remember, all antibodies are immunoglobulins, although not all immunoglobulins can function as an antibody. Okay, the main difference between the immunoglobulins and antibody is that immunoglobulin has a transmembrane domain in order to attach to the plasma membrane, where the antibody does have the transmembrane domain, whereas the antibody does not have any transmembrane domain. Now we will first discuss about the structure of antibodies and structure of immunoglobulins. The major structures of immunoglobulins are these. The FC region. FC region is the region where mainly the immune activation of immune system occur. The Fab region, where mainly the antibody binding sites. The antibody is mainly bind from in the Fab region, and antigen mainly bind in the FC region, which activate the immune system. Immunoglobulins also have some regions which is called as variable and constant region. Variable region is the unique for each antibody. Every antibody has unique variable regions, whereas constant region has constant for all antibody, but they are doing some various function, biological function in our body. Epitopes is nothing. The antigen determinant is called epitopes, and antigens mainly called it epitopes. Immunoglobulins also have some heavy chain and some light chain. Heavy chain actually classify the immunoglobulin, or we can say heavy chain can form the isotypes of immunoglobulins. Light chains are GABA and lambda. They are the two chains of light chain. Each immunoglobulins have any one of them light chain. This is the structure of immunoglobulin. So as I told you, four polypeptide chain, two heavy chain and two light chain form the Y-shaped immunoglobulins. They have this one is the Fab region, which consists of the light chain and heavy chain, the variable region of light chain, and the constant region of light and heavy chain. Similarly, this is called the Fab region, where mainly the antigen bind here on the N terminal or amine terminal. And this region is called the FC region, because this is the carboxyl and here the antibody mainly attach on the surface of antibody attach here on the FC region. So this is the terminals, the amine terminal, and this is the carboxyl terminal. So look at this. This is the heavy chain in the middle, and in the sides there are the light chains. The upper end is called the variable regions, and this lower part is all it's called as the constant region. They have a sequence of amino acids are constant, but they're doing a variable biological function. They are attached with disulfides bond. Each uh, constant region, heavy or light chain, is attached by the disulfides bond. The Fab region contain the variable light chain and heavy chain, and this variable chains consist of hypervariable regions. The hypervariable region is also called as complementary determination receptors or region, where different antigenicity can produce. This is called antigen binding site. This is called antigen binding site. And this area where this domain mainly attached to the antibody. This is the Fab region and this is the FC region. So as I told you, the immunoglobulins are Y-shaped molecules, four protein chains, two light chain, two heavy chains. What is FC region? This is the FC region. FC region is the region of antibody that interact with the cell surface receptors called FC receptors and some proteins of the complement system. They actually attach the some proteins of the complement system. This property allow the antibody to activate the immune system. The other part, the Fab part of the antibody is the main region of where the specific target antibody can bind. This pathogen attach on the Fab region. As I told you, the two variable regions, the variable regions and the constant region. The variable regions contain the NH terminals of light and heavy chains and is unique for each antibody with different binding sites. The constant region containing the carboxyl terminals of constant of all antibodies, but they are performing different biological activities, biological functions. And this is only the antigen binding sites, but they have a hypervariable region for different binding specificity or different antigens. Sorry. Now variable regions. The amino acids of variable regions vary greatly among the antibodies with different antigen specificity. It means this area is different for different antigen specificity. So that regions of variable heavy chain and variable light chain containing the hypervariable regions, they have a different antigen specificity, and that's why they are called hypervariable regions. And as I told you, it's complementary determinant region form the antigen binding site. What is constant region? As I told you, this part is called the constant region, the C part, the heavy chains mainly and the light chain. The constant region is the constant sequence beyond the variable region is called the constant region. The immunoglobulin constant region domain part in various biological functions that determined by amino acid sequence of each domain. So this is the heavy chain and light chain, the antigen binding site, the antibody binding site. This is the heavy chain, this is the light chain. The upper part is the variable region, this part is the a constant part. This is disulfide bond which links to each other, the constant region, the variable region. Antigen determinants or epitopes, the structure recognized by antibodies. Antibody actually they recognize the part which is called epitopes. Epitopes can bind in pockets or groove. It can bind the pockets or groove or an external surface in the binding side of the antibody. Now come to the functions of domain of immunoglobulins. So each immunoglobulins have a specific domain. This domain is called as the antigen binding domains. This part is called the FC region binding receptors. This part of the constant region is called complement binding site. And this part is called the placental transfer part. And this is mainly the immunoglobulin. G can pass the placenta. How we can subclass or isotypes of immunoglobulin. These isotypes of immunoglobulins are based on the heavy chain. Heavy chains, they are five distinct class of antibodies or immunoglobulin molecule according to the sequence of constant region of heavy chain. If heavy chain is mu, it is called as IgM. If heavy chain is gamma, it is called as IgG. If hemi chain is alpha, it is called as IgA. If heavy chain is delta, it is called as IgD. If heavy chain is epsilon, it is called as IgE. So according to their heavy chain, constant region of heavy chain determine the isotypes of different immunoglobulin, or we can say the subclass of immunoglobulin. Out of all these subclasses, the heavy chains of two immunoglobulins, IgA and IgG, have further classified on the basis of amino sequence and the different position and number of disulfide bond in alpha and the gamma heavy chain. So they are classified IgA1, IgA2, or gamma 1, gamma 2. Sorry, alpha 1 and alpha 2 is IgA1 and IG2, IgA2, and gamma is for IgG1, IgG2, IgG3, IgG4. So these two immunoglobulins can further classify on the basis of their different amino acid sequence and their position and number of disulfide bond. Now the light chains. In human, the 60% of light chain are GABA and 40% are lambda. Or always remember, each antibody has two identical heavy and light chain. A normal antibody molecule contains only one light chain, either the GABA or lambda, never both. Hinge region is the region where these two chains are joined to each other and form a one kink. So the gamma, delta and alpha heavy chain contain extended peptide sequence between the constant region 1 and constant region 2 that has no homology with the other domains. There are major four functions of antibodies or immunoglobulin. Number one, to neutralize the toxin and viruses. Number two, to opsonize the microbes so they are more easily phagocytosed. Number three, to activate the complement system. Number four, to prevent the attachment of microbes to mucosal surface. So this is the neutralization of pathogens. This is the complement activation systems. Antigen antibody complex activate the complement system and trigger its antibacterial activity. And opsonization is a process of phagocytosis cells grab the antibody bound to the surface of foreign substance for efficient phagocytosis. So engulf the process. This is opsonization. So these are the three major functions, and another the prevention of mucosal membrane through the mucosal surface through the IgA antibodies. So these are the immunoglobulin subclass or isotypes: IgA, IgG, IgM, IgE, IgD according to their heavy chains. Gamma, alpha, mu, epsilon, and D. The mnemonic is game D. These are the monomers of isotypes of immunoglobulins IgG, IgE, and IgD. These are the polymers, and IgM it's called as the pentamers, and IgA it's called as the dimers. Immunoglobulin G, the structure of immunoglobulin is monomers, and its percentage of serum antibody it's around 80%. The highest number of immunoglobulin present in our in our serum is immunoglobulin G, around 80%. It is mainly located in blood, lymph, and intestines. Its half-life is very high, 23 days. It has an ability of complement fixation. It can pass the placenta, and it is also called as a second immune response or the secondary antibodies or chronic. All the chronic infections are mainly containing this immunoglobulin G, and their major function enhance the phagocytosis, neutralize the toxin and viruses, protect fetus and newborns because it's past the placenta, and it has a complement activation. Some condition in which this immunoglobulin G is elevated, mainly the in chronic conditions, Infections of all chronic infections, hyperimmunizations, some of malnutrition, liver diseases, and decrease is mainly in the A gamma globinemias. Second immunoglobulin is IgA. It is called as a dimer, and these two immunoglobulins are attached with the chain which is called as J chain. And immunoglobulin A is also called as a secretory immunoglobulins. Always remember, it is called as a secretory immunoglobulins. Immunoglobulins A has a dimers as I told you, and it is a secretory immunoglobulins. Okay, and its percentage present in serum. This antibodies is mainly present around 10 to 15%. As I told you, it is called as a secretory immunoglobulins. They are mainly present in tears, saliva, colostrum, genitourinary tract, GIT, prostate, respiratory epithelium, bronchial tract, milk, blood, and lymph. Immunoglobulin A serum half-life is 6 days, and its role, as I told you, its major secretory immunoglobulins is mucosal immunity. Mucosal protection is the major role of immunoglobulin A. They cannot transfer the placenta, and they have not ability of complement fixation. Major function: localized protection of the mucosal surface, provide immunity to infant digestive tract. Mainly elevated in the macroglobulinemias and some of the infections, infectious mononucleosis and malarias. They are decreased in A gamma globulinemia. Immunoglobulins M is called as a pentamers, and its serum antibodies around 5 to 10%. They are located mainly in the blood, lymph nodes, and B cell surface. Immunoglobulin M, its half-life is around 5 days. They have an ability of complement fixations, but they cannot transfer the placenta. From mother, IgM cannot transfer into the fetus. But this immunoglobulins can produce itself by the fetus. Uterus can produce this immunoglobulin IgM, and neonates and newborns can have an active immunity. This immunoglobulins IgM is the first antibody. It's mean in any acute infection, the first immunoglobulins which is elevated is IgM, produced during an infection, effective against microorganism and agglutinating antigens. The other immunoglobulins are IgE and IgD. IgE is very specific for receptors on the mast cells, basophils, and eosinophils. They have a long half-life when cell-bound. They initiate a strong inflammatory reaction to parasites. They are also involved in allergic reactions. IgD is mainly present is an antigen receptors on the mature B cell. It's mainly present on the B cells. So immunoglobulin E, as I told you, is a monomers, and it's very low concentration in our body, but it's mainly present in the mast cells, basophils throughout the body. Half-life is 2 days. They have no ability to complement fixation or placental transfer. Their very known functions are, as I told you, all the allergic reactions. This immunoglobulin E is elevated, and it also elevate in the parasitic infections, and it recognizes the cancer cell as well. So it's increase in all the allergic reactions: asthma, hay fever, atopic skin disease, eczema, anaphylactic shock, and it's decrease in A gamma globulinemia. Mostly immunoglobulins are decreased in the condition of A gamma globulinemia. IgD is a monomers. It's mainly present in very small amount, 0.2%, and it's located mainly on the B cell surface receptors, and it can also be there in the bloods and lymph nodes. Half-life is 3 days. It doesn't have any property of complement fixation or placental transfer, whereas its main functions are it activate the B cells and defend body by immune systems, and it's activate to produce antimicrobial factors to participate in respiratory immune defense in human. It's increased mainly in chronic infections, This graph shows the primary response and secondary response. The immunoglobulin G is mainly produced in the secondary response in our body, whereas immunoglobulin N is mainly increased in the primary response. Antigen determinations on immunoglobulins. Three major categories of antigen determinations on immunoglobulin: the isotype determinations, the allotype determinations, and idiotype determinations. Isotype determination. Isotype determinants Isotypes are antigens determinants that characterize class and subclass of antibody. Isotypes are found in constant region of the heavy chain and the light chain. Isotypes are found in all organisms individual in the species. When an antibody from one species is injected into another species, suppose human antibodies is injected into mouse or mouse antibody injected into human, the isotype determinant will recognize as foreign antigen, inducing an antibody, produce an antibody response to this isotype determinant of the foreign antibody. So if one species and other species, something injected from one species, from human to mouse and human to another species, so the immunoglobulin isotypes mainly produce the antibodies, help to produce the antibodies. Antibody to isotypes are useful for quantitations of immunoglobulin classes and subclasses. Now, what is allotypes? Allotypes are additional antigenic feature of immunoglobulin that vary among individuals of same species. In isotypes, the two different species actually react and define their antigens as a foreign body when goes into one member of a species to another, whereas allotypes is the same individuals. If you give this blood of same individuals to one patient to another patient, then this allotypes actually is the antigenic features of immunoglobulin that vary among individuals. All the humans have different allotypes of same species and are have genetic control. Allotypes difference are localized to the constant region of the heavy and light chain. So isotypes and allotypes mainly present on the constant region of heavy and light chain. Allotypes actually differentiate, isotypes mainly differentiate the two species antigenicity, whereas allotypes differentiate in the same species, species antigens. This is the antigenic features. So allotypes, antibody of to allotype determinants can be produced by injecting antibody from one member of a species into the another member of same species who carry different allotype determination. That's recognized as a foreign body because they said this is not my, this is someone else antigens in our body of the same species. Allotype represents slightly difference in the amino acid sequence in heavy or light chain of different individuals. So allotypes mainly on the present on the amino acid sequence of heavy or light chain of the different individuals. Whereas idiotypes are localized on the variable region, not the constant region, whereas isotypes and allotype mainly present on the constant region. And this idiotypes is mainly present on the variable region. And what is idiotype? It's a unique antigenic determination present on the individual antibody of molecule or a molecule of identical specificity. Each individual antigenic determination of variable region is referred to as idiotypes. Each antibody will present multiple idiotypes. So idiotype determinants of the antibody can recognize by self immune system to generate